Hi, this is Maria Nadestad, and this is a detailed walkthrough of SplitThreader. If you haven't already seen the introduction video for SplitThreader, please do that first. This one goes more into detail on some of the aspects of SplitThreader in case you have questions as you go about using the application. And now a closer look at some variant signatures in SplitThreader. These are some of the variant signatures in SplitThreader, which I will use to illustrate how the variants are drawn in SplitThreader and how the copy number profile matches up with the strands of the variants. So this is an example that is a deletion here in pink. And you can see that the copy number profile is initially high, it drops down a little bit, and then it rises again. And that rise and fall matches with where these breakpoints are right here. So this is how a single variant is drawn. It has two breakpoints, which are right here and right here, which you can think of as kind of elbows, or maybe ankles. So these little lines right here, I think of these as feet. And so here the reads are mapping from the left to the right, and then right here at this little elbow point, they break off, and then the rest of the read maps over here, starting at this position and then continuing in the direction where the foot is pointing. And so that's what these variant calls mean right here. And you can see that that matches up nicely with the copy number because you have reads that are here, and then the copy number from those reads, the coverage of those reads decreases and it continues over here. So that's why you have this rise in copy number. So that should always be true, that when you have feet where the feet are pointing, the copy number should be higher. You can see the same thing for tandem duplications. The feet are pointing inwards and the copy number is higher on the inside than on the outside of this one. So again, you have two breakpoints here. The copy number rises the same place that the feet are pointing. We also see an inversion here, which is actually a reciprocal event. You can just barely see here that there's one I highlighted in black and then one in green. And the green variant actually is the reciprocal pair of the black variant. So these cancel out their copy numbers, which is why you can't see any kind of change in copy number in this region, because they have the same number of reads that go here, split over here, and continue this way, as come from one side, go over here, and continue in the other direction. So they cancel each other out, and this is a copy number neutral inversion. Another event is a translocation. So this is an interchromosomal event that goes from, in this case, chromosome 17 down to chromosome 8. So here we're showing a different chromosome on the top than on the bottom. These others I could show with just a single top view. So here the copy number is high and then it drops suddenly. Same as before, we could see the direction of the foot. The reads come from the left, they're split at this breakpoint, then they continue down here in chromosome 8 and go to the right where the copy number then increases. So reciprocal translocation is a special type of event that's made up of two different variants, and these are characterized by having opposite strands to each other, which enable them to cancel out each other's copy number, and these are usually part of a single event such that you have a sequence that has moved from one chromosome to another, and that sequence is small enough to not cause a visible copy number change at the resolution of copy number segmentation. And now let's take a closer look at the category matrix. This chart explains the variant neighborhood categories and the copy number concordance categories that SplitThreader finds. So the variant neighborhood categories are all based on where other variants are located relative to the one variant that we're currently focusing on. A simple variant is one where there are no other variants between the two breakpoints. This would be a simple deletion, for instance. So in this case, there are no other variants in between these two breakpoints, so we call it simple. And you can see that's true for all of these. And then the copy number concordance category has something to do with what the copy number is doing here, but it has no influence on what other rearrangement variant calls are nearby. It only has to do with the copy number. So this is simple. A reciprocal variant is one that has another variant with the exact opposite strands that are within 10 kb of where this focused variant is. And so here we show the other variant in red. The variant that we have chosen here is that we're focusing on is shown in black. Because they have opposite strands and they're within a short distance of each other, that makes them reciprocal. The 10 kb is a threshold that you can change yourself, but 10 kb I found in my own study to be a good measure for capturing most of these reciprocal variants without including too many of just very crowded variants that happened to have others nearby that had the opposite strands, but that were not actually particularly closely related. Another type of variant is solo, which basically just means that it's alone. The variant calls are not nearby any other variant calls, and the threshold here is 100 kb. So this means that these variants on both breakpoints, there can't be any other variants within 100 kb of them. And that happens fairly often unless the cancer genome is extremely rearranged in that one region. And this is particularly useful when you start to look at the copy number concordance categories, because when a variant is this far away from other things, you should be able to expect matching copy numbers. And so that's really a common theme here, and why I show that the variant neighborhoods and the copy number concordance should be considered together. That's why I draw a matrix, so that you can see how they fit together and use it as a starting point for manual curation. Crowded variants are simply variants that don't fit into any of these other types, and it means that there's other variants nearby. This can be as little as just one other variant that's within 100 kb of one of its breakpoints. Here I show a more common case where there are several other variants nearby. But it can just be anything where it's not alone, it's not reciprocal, and it's not a simple variant. So for the copy number concordance category, matching means that both breakpoints of the variant have a matching copy number change. Here it means you have this decrease right here, you have an increase over here, it matches with how the feet are located, and you see the same thing for these other types over here. Partial means that only one of the two breakpoints has a matching copy number, but it does have to be matching in the correct direction and everything. And that's again the same for all of these. It's regardless of the variant neighborhood category. It just means what is the copy number doing around these two breakpoints. The non-matching means that at least one of them has the wrong exact opposite copy number of what we would expect. In this case, you can see the feet and the rise in copy number are on opposite sides, so that could potentially point to some issues in variant calling. If you see this happen a lot, it could be that we're interpreting the strands incorrectly. If it happens, more often than having actual matching copy numbers, for instance, then that should be a warning sign. So that's why we've included this particular type. We also get variants that may be copy number neutral. In the case of reciprocal variants, this is actually expected because the strands of these variants would cancel each other out, and so you're not expecting to see a copy number change here if the two variants have about the same number of reads that are then crisscrossing each other and canceling each other out in copy number effect. This is not at all expected when you see a solo variant, for instance, because you don't have any other variants that can explain why the copy number is not showing up. And so this is another kind of warning sign. Simple variants you have to be careful with. Sometimes the simple variants end up being so small that the copy number change is not visible at the resolution that we have segmentation. If the variant is above 100 kb in size or more, that's not something to worry about, but anything below that, and you might be missing it because the copy number segmentation could be just going right over it, thinking of it as an outlier. And that's a common problem with any type of copy number segmentation. And the last type here is if the neutral variant is also crowded, then you don't see that as a warning sign because there are other variants nearby that could be canceling out its copy number. And so it can be really hard to tell whether this is particularly an issue or not. So I wouldn't call this one a warning sign. Some of the variants that you would most trust is any kind of reciprocal variant. The fact that there are two of them makes it kind of an interesting variant type. It lends extra confidence to one variant when there's exactly another one that is the opposite of it. They kind of serve to reinforce each other. Simple and solo variants, any variants with matching copy number is probably very good. And the worst warning signs I can see are when a solo variant is completely alone, has no copy number change whatsoever. That would be a warning sign that the variant may not be correct and that it's a good thing to look at in further manual curation. Go and see whether there's real evidence for this variant or whether it could be some kind of artifact based on repetitive elements and other issues with variant calling. So that's the variant neighborhood category and the copy number concordance category, and what you might conclude from the intersections of these two variant categorization schemes. So now I'm going to do a demo and show you how to use SplitThreader. This is a bit more detailed than the introduction video, but there is a bit of overlap. And this one's just going to go into more detail on each of the tabs and settings, parameters that you can set along the way. And so let's get started. This is SplitThreader.com. It's a web app for exploring rearrangements and copy number amplifications in a cancer genome. In order to use SplitThreader, you have to upload a set of variant calls and a copy number profile. The variant calls you can get from running sniffles on long-read datasets or lumpy or another structural variant caller on a short-read dataset such as paired and Illumina data. The copy number profile you can get from running Copycat. There's a link right here. If you open that up, it'll send you to my GitHub page where there's a quick little script here, shows you how to use it, and you're going to end up with a neat little copy number profile that you can then upload here. Then you type in a description of your data set. So I'm going to do this right now. I have a little demo page here. Let's see, variant calls. That's my bedpe file. You can use bedpe or a VCF file. The instructions are right over here. Tell you how to do that. And I'm also going to upload my copy number profile, which I got out of Copycat from that GitHub link. And then I'm going to type in a description of the data set. In this case, these are variants from the SKBR3 cell line. I have a number of different variant call sets, perhaps, so I'll say that they're sniffles variant calls. I can also say SKBR3 breast cancer sniffles call set. And I'm going to then submit that. It'll say, great, both files were uploaded. That's a quick check to make sure the server is working correctly. And so it reads the input files. It noticed that it was a sniffles bedpe file on its own, and it parsed it accordingly. And then it filtered out a bunch of small variants here, because sniffles will call things down to fairly small variants. And in SplitThreader, we're really focusing on variants that are above 1 KB in size. That's the current filter on the back end. Anything below 1 KB, it's not going to show you because those things are really better viewed in IGV at this point. SplitThreader is designed for long-range variant calls. So now we just wait for it to finish segmenting the copy number. This usually takes a few minutes. I don't know that I've ever seen it take more than 10 minutes, and this is because we're using 10 KB bins. If we used much smaller bins than that, it would take a longer time, and it would be a lot more data to show, but we're sticking with 10 KB bins. That's currently the only thing that SplitThreader supports, and it's the only thing that Copycat will output. So now SplitThreader has finished. the segmentation of the copy number is done, and it now shows up with this big button here saying Show Visualizer. So you can see if anything went wrong down here. It'll show you if there's any issues or any warnings about your input data sets. And we're going to click on Show Visualizer. So now this shows us all of the variants that are more than 1 KB in size. You can see it's pretty dense here. What we can do on this view is we can see copy number profiles here, and this is the segmented copy number profile. It starts by showing you chromosome 1 and chromosome 2 at the bottom. If we scroll down a little bit, you can see chromosome 2 down here. And there's a number of different things we can do. We can go to different chromosomes. I can, for instance, show you what chromosome 20 looks like by dragging chromosome 20 label from here on the circles plot onto one of these plots. I can also show you what chromosome X looks like down here. And you can see the copy number profile, you can see connections. One of my favorite regions is the HER2 region, which is properly known as ERBB2. It starts by showing the HG19 gen code annotation. We also have GRCH38, so you can pick whichever one works for your data set. If you have another annotation that you want to use, just put a comment on this YouTube video and I will address it. I'll get the annotation on there that you want. Just send me a link. And what we can then do, this is where chromosome 17 is showing ERBB2. We can zoom out. This is the beautiful HER2 region in this genome. It's very nicely amplified, even though it's very nicely amplified, but there's a baseline copy number on either side that's very stable. This is an interesting region. If we pull out chromosome 8 here, you can see all these connections were to chromosome 8. If you're curious about what a connection might be, such as maybe one of these down here, you can click on it, and if we click on a variant, it's gonna go to the variant tab right here, and it'll show us some detail on that particular variant, and it'll zoom in on the variant so we can see what it is. And it gives you the variant name, so you can look it up in your original input files if you need to. See what chromosomes are involved. You're zooming in, you can see the CNV category and reciprocal categories and so on. And yeah, so this is actually a reciprocal variant, which is pretty cool. So to walk around here, you can explore genes. You can even show all of the protein coding genes in a region. So if I put those in, I'm showing any protein coding genes that are present right here. You can see there's a couple of them. You can also show pseudogenes, anything you're interested in particular here. MicroRNAs, none of those. Some antisense, link RNAs. And if there's way too many of them, like if you zoom out a lot and it gets confusing, you can also hide the gene names and just get an overview. So that's some things you can do on genes. There's also variants. If we say we zoom out to show we'll go back to chromosome 1, chromosome 2, and you can see there's a lot of deletions here. What would happen if we removed all of the deletions? Wow, a lot of them go away. If we only show deletions, this is what happens. And so you can just get an idea of the variant types. You can also hover over any variants to see what they are. Inversions, interchromosomal, BND is a break end, which is basically like a breakpoint from a translocation of some kind. Usually to different chromosomes. And again, you can see variant details. There's also all sorts of fun stuff here. If you've zoomed in on a particular region that you find really fascinating, and you just must know what's going on here, and then you can it'll show you exactly what the coordinates are here, and if you click on one of these, it'll take you straight to the UCSC genome browser for those coordinates so that you can see what's going on here. And yeah, it takes you to the right chromosome, the right coordinates, and you can see what's going on in this region using whatever tracks in the UCSC genome browser you're most interested in. And it'll automatically show you either GRCH 37 or 38, depending on which annotation you're looking at. And yeah, change the database and genes tab under annotation. So that's going to change which genome you're looking at in UCSC. You can also send the variants to Ribbon. You can send all of them. Ribbon is a visualizer that I've made. It focuses on making it easier to look at long read alignments, but it'll also work for short reads. So if you want to take some of the variants you're interested in, export them to Ribbon, and you can load a BAM file and see what the alignments look like around those variants. We can also change from showing segmented coverage to showing just regularly unsegmented coverage. So if I click this right now, you're going to see that this region ends up showing a lot more of the detail of what the original copy numbers were. Yeah. You see there's a giant outlier right here that just increases the level like crazy. That's some of what segmentation protects us from. Oh yeah, it took a second too to load the bottom one, and I've zoomed in on the top, so you can see it's very coarse here compared to the segmented level that's basically just very flat. The reason I show segmented coverage by default is that it's faster to load, and so that way we can load coverage for all the chromosomes at once, and it's faster to interact with it. But if you want to see the extra detail, then all you have to do is uncheck this box. If you know or have decided what your baseline copy number might be, let's say we think the average copy number for a haploid chromosome in this case would be 14. And so the diploid level is 28. So I might divide the coverage by 14, hit enter, and then it's going to show me the scaled coverage here. So it's just going to scale the y-axis accordingly. So then I can see, okay, I'm at about a level of 3 copies of DNA here. And you can filter variants in a lot of different ways. In this particular data set, it's with sniffles, it's long reads, so we're using split reads to do variant calling, but there's no paired end reads. I can use the variant size here to filter as well. So there's variant size, number of split reads, number of discordant pairs, and some miscellaneous read evidence. These 3 here come out of the VCF file, so it's going to say like SR equals or PE equals for this, or it's going to give you some other strand information that's not divided into split and discordant pairs. And so whatever I find in that VCF file as I'm parsing it is going to go in here. And the way you can check what you're looking at in that case is to go into the variant analysis tab and just look at your different variants, see what they have here. Split is the number of split reads I found, number of pairs, so there's nothing here because it's long reads, and any other read support that you may have found in that VCF file. So you can see what's going on there. And as we go back to the visualizer, let's filter. Right now I'm showing a minimum of 1 KB in size, and if I want to filter to a minimum of 10 KB, then that's going to clear up this plot a whole lot. Yeah, so I can also remove that again. Now we're showing all the variants. You can especially see it here at the bottom. There's just a lot going on. So we really can clean it up a little bit by not showing the tiny variants. And you can also filter for number of split reads. Here we already have a minimum of, I think, 10. So if I say I need to have 20 split reads before I believe this variant, maybe I have higher coverage or something, then this is all I'm left with. And so you can just do this kind of filtering to see more of what's going on, do some filtering on the fly. And yeah, that should be pretty useful. You can download the coverage data and the variant data. They both end up as CSV files because it's how I'm storing them. And this is something you can very easily load into Excel and then start annotating and seeing what kind of variants you think are interesting or what's going on with the segmented coverage. So you can put that into another, like IGV or something, put in segmented coverage there too. And that's all I wanted to show you for what you can do with your own data. Thank you very much.